And if I reverse, you can see it. You can see both of them changing their shape. As you can see, and I don't even know what this type of cell is, but if you guys can like let me know what it is in the comments, I'll highly appreciate if you tell me what this type of cell is in the comments.
Hey guys, just to let you know that I have a Patreon.
Nice.
If you are interested in supporting me, head on over to patreon.com and subscribe for some exclusive content.
Hello, hello, ladies and gentlemen. It is time. What we're going to be doing today, ladies and gentlemen, is we are... oh, sorry. There you go. We're going to be doing a science stream about hematology. Basically, I haven't done this in a long while. What we're going to cover here today is everything that's part of the erythrocytes. What we're not going to be covering today is, if I'm not mistaken, the coagulation process. I'll probably save that for the platelets if I ever do a video on it, or a stream basically on it. But aside from that, what we're going to be doing today is going over what red blood cells do, and their only function. That's what I said. They're only good at one thing, is to deliver oxygen throughout the body. And obviously, oxygen is essential for life. We move on to the hematocrit. Hematocrit is basically like the level of blood you have in the body. More specifically, the ratio of red blood cell to the plasma. If we start off with the plasma, which is the yellow part at the top, 55% of your blood in general is composed of the plasma. The plasma, which is 90 to 95%, is made out of water. As I've stated before in the last slide, you have plasma proteins that are existing in the plasma. Then you got molecules and electrolytes that are also circulating in the plasma. Nutrients such as glucose and amino acids, enzymes, hormones, metabolic wastes such as CO2, if I'm not mistaken, urea or urobilinogen. I get confused between both of them. I'm going to have to get corrected on that, but that's also metabolic waste as well. And plasma also functions as a transporter. We move on to 1%, and this is really interesting. 1% is your leukoplatelet layer. Leukoplatelet meaning your white blood cells. Such as macrophages, neutrophils, eosinophils, basophils, and etc. And your platelets as well. And finally, the rest, 45% of it, are your erythrocytes. We move on finally to the erythrocyte itself. We're going to look at the anatomy right now. First thing we have is the plasma membrane. This whole thing over here, sorry about that. This whole thing over here is the plasma membrane. Like I said, they're anuclear. They don't have any organelles. What they do have inside of them, because they eject their organelles once they get mature, what you're going to find inside of them is just a bunch of what you have here, which is the hemoglobin, made out of B protein chains and alpha protein chains. I should have actually precised. I should have went a little bit into precision that this is hemoglobin right here. This whole thing is made out of hemoglobin, and it has a beta protein, 2 beta protein right here and here, and 2 alpha protein chains, which are here and here. I don't know why I didn't precise that. I'm so sorry about that. Anyways, moving on. In between, right here, here, and here, and here, there's always going to be 4 spots available for the heme group. And the heme group, that's the most important part. That's where the oxygen binds onto. The oxygen is always going to bind to the iron that is in the middle of the heme group. And you have its composition over here. I'm not going to go into much detail. This is not really a chemistry video. But the oxygen, the O2, is always going to be attached at the middle of this heme group. The structure of globin protein, the heme group, and also as well, we're going to get into the structure of the ankyrin and spectrin protein, which allows the red blood cell flexibility. The ankyrin protein is, actually, if I'm not mistaken, ankyrin is between, like, right here on the edge of the plasma membrane, while the spectrin keeps it in place like this, and they're both, like, attached to each other, which allows the erythrocyte more flexibility. Finally, there's antigens A and B, actually either A or B or both, and or none at all, that can be present around the membrane of the red blood cell. And that's actually going to be our next slide. We're going to talk about the different types of blood types right before we get into the life of the red blood cell. Now we get into blood typing right over here. Blood typing is, in a nutshell, what the, it's basically labeling blood, if I can put it like that. As you can see, you have different types of, blood here. You have the A type, the AB type. Actually, it shouldn't be even be, it should just be B. So yeah, you just have the A type. I just recognized I have a mistake. Don't worry about that. You got the A type, the B type, the AB type, or the O type. And on top of that, you also got another another antigen, which is the Rhesus group. The Rhesus group is the plus or minus. Plus or minus means you either have it or you don't. Meaning that, for example, if you are A positive, it means that you have a Rhesus antigen on top of that. Meaning that you can only receive negative blood because you don't have the positive Rhesus group. And that's really important because there are some blood pathologies that we're going to get to at the end that do have that as well. So that's basically the Rhesus group. The problem with the Rhesus group is that if you are A plus positive, there's no problem. You can still receive A positive blood. You can still receive A negative blood. You won't have any antigens, anti-Rhesus antibodies inside of you, right? Because you're A positive. The problem is when you give to the negative. The moment that you mismatch and you give a Rhesus positive to a negative, this guy's going to start developing antibodies to the negative, to the positive Rhesus blood that he's receiving. And that causes a problem because when those antibodies are in the blood, you can't give A positive blood anymore. And the problem with that is that the blood starts coagulating. Now, as I was saying here, there may also be antibodies present in the blood opposite to the other type. So, for example, if you're the A type, you're going to have, in return, B antibodies circulating in the blood. That way, to prevent intrusive blood that is not of your type when you're getting a blood transfusion. And that's really important to never mismatch these, because once you receive mismatched blood, it's an immediate risk of death because the blood starts to clump together. The antibodies begin to aggregate the red blood cells together. And that creates embolism, that creates blocks, which leads to eventual death. In the beginning of time, to get to a healthy red blood cell that has a lifespan between 100 to 120 days, it all starts in the spongy red bone marrow. Any red spongy bone marrow. Specifically, this one that I took was the femur, but there's also other parts of the body where you can find spongy red bone marrows. That is where red blood cells are created. What happens is you're always going to start with a hematopoietic stem cell, or a pluripotent stem cell, however you want to call it. In any case, this guy has two pathways when he divides. He can either divide into a myeloid stem cell or the lymphoid cell pathway. The lymphoid cell pathway is not what we're going to be getting into. We're going to be getting into this pathway over here, the myeloid stem cell pathway. And now, once your pluripotent stem cell or your hematopoietic stem cell gets to the myeloid stem cell pathway, now the myeloid stem cell has three positions to divide into: either platelets, red blood cells, or white blood cells. Now the question is how do you get it to choose our target red blood cell? Well, that is where you have to follow this arrow over here, which leads us to the nephron cell. The nephron cell is the nephron is actually I should have actually put here in parentheses, this is the kidneys. Actually I should have said nephron not available. It's always available. How can I put this? Nephron inside the kidneys. Yeah. So this is usually a nephron that is gonna synthesize the what we're looking for is EPO, which stands for erythropoietin, which is available in these cells, in these nephron cells. Usually there's going to be a protein that is usually binds on the I think it's hematopoietic intrinsic factor. I'm just going to make sure that I'm not spewing any BS. Hypoxia induction factor. I was right a little bit about a few words. Hypoxia or hematopoietic inducing factor. But anyways, there's usually a protein that binds onto it because you don't want to let it run loose and always synthesize the EPO to get red blood cells. Because what can happen is that if you have too much EPO, and we're going to get into a pathology later on for that, is that if you have too much EPO going on, that creates too much red blood cells. Too much red blood cells lead to an increase in viscosity, which is really dangerous, and you don't want that. So there's always going to be this protein that regulates the hypoxia inducing factor to not run wild, because it always is going to want, it has a high affinity to bind on, to go inside of the nucleus. It always wants to go inside of the nucleus and bind on to the EPO gene to increase the production. So it always wants to increase the production of EPO, which gets secreted into the bloodstream and eventually is going to make its way to the myeloid stem cell, stimulating it into a pro-erythroblast. Now, a pro-erythroblast means before the red blood cell. And this is where we need to basically order building blocks to finally arrive to our mature erythrocyte right here. Now, a way to do that is by intaking food. Nutrients is what we need. We need to order these stuff, right? By our food intake. And now we're going to make our way into the stomach. And the building blocks that we're going to order, hashtag courtesy of Amazon, is, first of all, iron. Like I said, the heme part is essential of having an iron group inside of it, but not the iron 3+. That's very specific. We also need B12. We need the iron 2+, not the 3+, and we're gonna see how it changes into 2+ form so that it binds onto the heme group. B12, vitamin B12, very essential for the maturation, for the maturing of the red blood cell. Folic acid as well for the maturing factor of the red blood cell, and then the rest is just amino acid, carbohydrates, and then fatty acids as well, usually found in foods. I feel like this, let's see which one is the most simplest. Let's see which one is the most simplest. I think it's going to be the folic acid. Yeah. Let's start with the folic acid at first. Folic acid usually gets intaked. Usually it was going to make its way through the stomach, goes inside of the intestinal tract, the duodenum, the jejunum, and etc. You know the rest. And it's gonna travel towards, eventually it's going to meet up with these cells. What are these cells? These cells are your enterocytes. The enterocytes have this specific protein channel that is specific to the folic acid. It intakes, and it's usually a simple pathway. It's just gonna like intake it. It's gonna then eject it into the blood circulation. Very simple. And then finally, the folic acid makes its way into either two sites, either to the erythroblast or the polychromatic erythroblast to stimulate its growth. That's what it simply does. It's very simple, very straightforward. You don't need to understand a lot more about the effect about the pathway of folic acid. Same thing for B12, if I'm not mistaken. B12, vitamin B12 needs just one thing to travel because it doesn't have, it can't travel on its own. It needs help. And that's where these guys come in, your parietal stomach cells, which are found in the stomach right over here. They're found actually all around, but for the purpose of this presentation, they're just right over here. They secrete something called intrinsic factor. And the intrinsic factor, once B12 gets inside of the stomach, it's going to bind on to the intrinsic factor, creating this complex, making it able to travel. It travels all the way inside of the intestinal tract, making its way to another enterocyte. Once it makes on its way to the enterocyte, the only difference is that now this protein binds on to the intrinsic factor receptor, which is this guy, found on the enterocyte's plasma membrane. Then it's going to get endocytosed, and then it's going to make its way into the membrane where it's going to fuse with it, and it's going to exocytose out into the bloodstream. Once it's inside of the bloodstream, there's another protein that helps it travel all the way to the spongy red bone marrow, and that protein is called Transcobalamin, if you want to know about it a little bit more. But it's still going to bind onto it, create the complex, and again, it's either going to go to the erythroblast over here, still stimulating its growth, or it can also go at the same time to the polychromatic erythroblast, stimulating again its growth. That's very simple and straightforward. Its only difference is that it just needs a protein to help it travel. That's pretty much it. Finally, we get to our iron. Iron is a little bit more special. Iron basically needs to... same thing. It travels inside the stomach, makes its way all the way to the intestinal tract. Once it's inside the intestinal tract, it's going to travel to our enterocytes again. As you can see, it travels to the enterocytes. But here's where the magic happens, and this is the important part that you need to remember. First, it has to get converted into iron 2+, in its 2+ form. And the way it does that is that there's a specific enzyme on the enterocyte's plasma membrane. It's called duodenal cytochrome B enzyme, which converts iron 3+ into its 2+ form. And now it's able to travel inside by its plasma protein channel, which is called the divalent metal transporter type 1. It's basically going to intake iron 2+ inside of the cell, and at the same time, it's going to also intake a positive proton alongside with it. Pretty simple, right, so far? And once it's inside of the enterocyte, there's another protein called ferritin, which has a high affinity for the iron 2+ form. They're both going to bind together, and they're going to make ferroportin. And now there's two pathways. What's really interesting is that the first pathway, which is can combine with other ferroportins, and together they're going to make hemosiderin. Now, why am I telling you this? The reason why I'm telling you this is because hemosiderin is like, think of it as like storage, iron storage. It's going to store iron inside of it, and when it needs it for more erythropoiesis or hematopoiesis, however you want to call it, it's gonna make it travel outside of it, inside of the bloodstream again, to make more red blood cells. So hemosiderin is just essentially like iron storage. That's what it's basically is in a nutshell. Correct me if I'm wrong again. Now, the second pathway, which is what interests us, because we want to get to our mature red blood cell, is directly traveling outside of the red blood cell through the ferroportin transporter. It's another protein channel which ferroportin travels through easily. Once it's outside of the interstitial space, it's going to get transformed by hephaestine, which is an enzyme that converts iron 2+ into 3+ again. I don't know why it does that specifically. I don't remember why, but it reconverts it back into iron 3+. To basically send it back into the bloodstream. Maybe because in its iron 2+ shape, there's a lot of affinity for other proteins for it, and you don't want that. You want it for your iron 3+ to get into the spongy red bone marrow, so that's why they convert it again. Maybe, I'm not sure. But again, correct me and add more information in the comments. Please do add more information. I really love that when you add more information in the comments regarding this type of stuff. It really makes me learn a lot more than I spew out, essentially. So once your iron 3+ goes in size and passes into the capillaries, it binds on with transferrin. And transferrin is going to help the iron 3+ for the Heme Group, where it goes back again inside either two pathways for the maturation of both of either the polychromatic erythroblast or the erythroblast itself. And finally, there's one more a few more substances, but we consider them as one. I put them into one pathway. It's your amino acids, carbohydrates, and fatty acids. These are your three last substances that are needed to mature. These are pretty much straightforward. These guys just follow again the stomach. They get intake. They get absorbed. They just get absorbed through digestion directly into the blood vessels. You find themselves inside of the blood vessels, and again, they can go to either two pathways, to the maturation of the erythroblast or your polychromatic erythroblast. And finally, I've been talking about these two these two guys, the erythroblast or their polychromatic erythroblast. The difference between these two is that your polychromatic begins having this red pigmentation on top of it, as you can see here by these like red dots. Once all of these guys, all of these substances, your B12, your folic acid, your amino acids, and etc., your iron, all get inside of the spongy red bone marrow and mature, help the growth of your red blood cell. Finally, it goes into an orthochromatic erythroblast. The red pigmentation starts taking over and more over space eventually, because what's happening is that you're synthesizing not only heme, but you're synthesizing globin, and together that makes hemoglobin as well. And you're synthesizing so much that it starts taking so much space, and that's where the ejection of the organelles, including the DNA, sorry, not the DNA, but the nuclei, gets taken out, gets ejected out. Your mitochondria, your Golgi apparatus, any organelles basically. And what happens to them, they just don't stay there. As you can see here, they get digested by your macrophage, and they get digested and turned into proteins as well for even more synthesis of either red blood cells, platelets, or white blood cells, or any other proteins needed for the synthesis of your leukocytes, or any other cells that get synthesized inside of the red bone marrow, basically. Finally, once you get to the orthochromatic erythroblast, it is finally able to be ejected out into the bloodstream from the spongy red bone marrow and finally mature into our erythrocyte. After 100 to 120 days, about 100 to 120 days, it becomes old, it becomes decrepit, and eventually it gets taken in by a macrophage. Now, the question, I didn't precise here on this slide, but usually the death of the red blood cell happens wherever you have like sinusoidal capillaries. Sinusoidal capillaries meaning that they're spaced in between themselves. And if they get stuck in there, that's where your macrophage comes in and phagocytoses the red blood cell. Usually that your sinusoidal capillaries are found in your liver, your spleen. The spleen is actually nicknamed the graveyard of the red blood cell, actually, because that's where they go on to die. Your liver as well, and anywhere else where you have sinusoidal capillaries, basically. So your macrophage phagocytoses the red blood cell and then starts digesting it, and into two pathways. One of them is your heme group, and then the other one is your globin group. I'll start with the globin group because it's really straightforward to explain. Your globin group just essentially gets digested back into their separate alpha chain and beta chain into amino acids, basically. That's it, and then they get ejected into the bloodstream for more recycling into the synthesis of either red blood cells or any other. Actually, no, red blood cells for that I'm not gonna. Actually, no, they're just amino acids, so they can definitely get recycled back for other stuff as well. We get into the heme group afterwards, the heme pathway. Now, the heme pathway gets digested into two paths. I'm going to go into the iron group. Now, again, macrophages also have inside of them, they have this blue protein, which is ferritin. And you know the story, ferritin really likes binding with the iron 2+ group. And then once it does, it creates ferritin. And ferritin has two pathways. Ferritin can either go create more hemosiderin, where it stores the iron inside of the cell, or it goes into the ferroportin transport protein channel outside, gets released into the bloodstream, for the next red blood cell synthesis, basically. Finally, there's the other part of the heme group, which gets digested into biliverdin. Now, biliverdin needs to be converted into bilirubin via the enzyme biliverdin reductase, which is an enzyme which creates unconjugated bilirubin. And then this unconjugated bilirubin gets released into the bloodstream. And now you're probably wondering, what in the heck is unconjugated bilirubin? Well, unconjugated bilirubin is it's a substance used for a lot of things. Mainly, it's basically metabolic waste, if I'm not mistaken. If I'm mistaken, it's also like, because I think, I don't know, it is metabolic waste essentially. And what it does is it binds onto the albumin, because it can't transport it by itself, so it needs help from a transport protein. The transport protein brings it into the liver, where it unbinds from it, and the albumin just goes back into the bloodstream, and it gets recycled, vice versa. It's not really important that much. It's just a transport protein. Finally, the bilirubin, the unconjugated, gets conjugated with glucuronic acid, and together they make conjugated bilirubin inside of the bile. Now that bile liquid travels through the duct inside of goes through the pancreas back into the digestive tract. And as we follow the digestive tract, there's your microbiota bacteria that breaks down the conjugated bilirubin into urobilinogen. And at the beginning, I talked about that those were metabolic wastes, and the metabolic waste is that urobilinogen. 10% gets reabsorbed into the bloodstream towards the kidneys, and then the rest mutates into stercobilin. Give me a few seconds. I'll just go back to the urobilinogen. It goes back into the kidneys to get synthesized as urea. That's the pigment of why your urine, what makes your urine yellow. And then finally, stercobilin is the fecal pigment of your feces, basically. It's what makes it brown, basically. So, all of that to say that this was hematopoiesis. Moving on now to the transfer of the Bohr effect and the Haldane effect. It's, at the beginning I said, one of the main functions of a red blood cell is the transfer of the transport of oxygen to the tissues, and the transport of CO2 outside of the body. So, first of all, we're going to start with the Haldane effect. Usually, the way that it's explained is that the Haldane effect triggers the Bohr effect, and the Bohr effect triggers the Haldane effect. But usually, what they do is that they separate it in two pieces. The Haldane effect is more prominent inside of the alveoli. The Haldane effect means where the O2 binds on to the heme group, and the CO2 leaves the red blood cell back into the alveoli. When you breathe, you intake oxygen while you intake air, but some of that air has oxygen in it. That oxygen, once it gets into the alveoli, has a pressure of 104 millimeters of mercury. By the law of diffusion will always go where there's a higher concentration of pressure to the lowest concentration of pressure, as you can see here. The problem is that O2 cannot bind on to the heme part just yet. it needs space. And that's where your CO2 needs to leave. And the way it does that, the first part that it does, and I'm going to start with the longest one at this point. There is CO2 that gets binded on, that is binded on here, here, a little bit of everywhere. It doesn't bind to the heme. CO2 is never binded onto the heme. It binds onto the globin group, to the globin proteins. So it binds on here, here, and here. So the CO2, again, the partial pressure of CO2 inside of the pulmonary capillaries is at 45 millimeters of mercury. Again, the law of diffusion states that it will go from higher pressure to lower pressure, so from 45 millimeters to 40. And it senses that. So what happens is that CO2 starts detaching. Eventually, 20% of the CO2 that was attached to the globin group leaves and goes into the alveoli. Now there's space. Now we're beginning to have a little bit of space for the O2 to bind on to the heme group right here. But there's a little bit more of that, and that's where the second pathway comes in. It's the conversion of bicarbonate back into its CO2 form. And what happens is the bicarb starts entering the cell, because what happens is the pH starts increasing. The pH starts increasing because of the CO2 that starts to leave, right? And what happens is that your chloride ions start to exchange with this protein membrane. It's basically, in a nutshell, called the hamburger effect. It exchanges bicarbonate with chloride ions, so that the bicarbonate, alongside with the hydrogen protein, well, the proton ions, I'm looking for the specific word here. I have a blank. My memory is blank right now. They go together to produce H2CO3. Now, before going a little bit further, I'll just want to explain that on the globin group, you also have as well positive protons that also contribute to the pH. But these guys contribute to lowering the pH. Now what happens is that once HCO3 comes inside of the red blood cell, these guys love and are attracted to the HCO3. And they want to combine together to create H2CO3. And what happens when you do that, that also increases the pH to a more alkaline state. And what happens, your H2CO3 eventually, alongside of carbonic anhydrase, converts the H2CO3 and reacts with the carbonic anhydrase and produces water and your CO2. And 70% of that CO2 that was inside of its bicarbonate form goes back and diffuses right into the alveoli. And now finally we have that space available for the oxygen to bind on to the heme group right here. And there's always going to be 4 spots. The heme group always has 4 spots on the globin, making hemoglobin as well. And also as well, it's also important to talk about this, but the rest is that 2 to 10% of the CO2, which is inside of the plasma, which diffused inside of the plasma and mixed with the plasma. I don't know. There's a specific word for that, but I can't really remember right now. But 2 to 10% of the CO2 available in the plasma diffuses right into the alveoli. Finally, there's 2,3-DPG. 2,3-DPG stands for 2,3-diphosphoglycerate, which is a byproduct of cellular respiration. I'm going to get into that as well. This guy also unbinds because if the pH goes up, it will unbind because there's no more CO2 to keep in place, and it will just go back into the circulation waiting for the next red blood cell to bind onto it. But we're going to get into that when we do the Bohr effect. Now, we finally have our O2 ready to go all the way to the alveoli, which brings us to the Bohr effect. Now, once we're inside, now back from the alveoli, the red blood cell circulated into the circulation and arrived at the tissues level. And again, same principle applies. The partial pressure of O2 is at 40 milligrams of mercury and CO2 is at 45. And this is where the inverse happens. The O2 senses that. So it starts preparing to leave. And the way it does that is that this high affinity begins with cellular respiration. Now, cellular respiration is going to start converting glucose and oxygen that it intakes all the way into 6 oxygen, 6 water, ATP, and as well as heat. Now, the heat is not that important, but it's also it's still a component of the cellular respiration. It takes the heat. It basically takes the heat, and then the oxygen starts feeling that and begins unbinding. But that's not enough. That's what I'm trying to say. That's not enough. Another thing that's going to start happening is the CO2 that's produced. That CO2, first of all, 20%, 2 to 10% gets diffused and it gets dissolved. It gets dissolved. 2 to 10% gets dissolved inside of the plasma. Again, the diffusion laws of the pressure, the CO2 goes inside of the capillaries. Now, the second part is the 70% that needs to go inside of the red blood cell and that needs to get transformed back into its bicarbonate form. And the way that happens is basically that CO2 mixes in with the water available inside of the red blood cell. Carbonic anhydrase helps stimulate that process back into H2CO3. The H2CO3 gets converted into bicarbonate and into positive proton. And this is where the interesting and fun part becomes, is that if you start producing a lot of these protons, the pH lowers, making it into a more acidic state. And this is where it highly contributes to the O2 beginning to unbind, because the O2 does not like an acidic state. It starts unbinding and going into the tissues because these proton ions begin latching on to the globin group, and that lowers the pH, which brings us to this this equation right over here, this molecular equation, which reacts positive proton ions react with the oxygenated hemoglobin, which creates deoxyhemoglobin and 4 oxygens being able to go inside of the tissues, which contributes more to the cellular respiration process. With that being said, now that there's enough space, the hemoglobin will be in its what we call its R state, which stands for released state, if I'm not mistaken. Actually, no, it's in its T state. Sorry about that. Forget what I just said. It's in its T state. I don't remember what the T stands for, but it's in its T state. Again, correct me if I'm wrong, but now, what that basically means is that the CO2 has enough space to go latch on to the globin group, as you can see here, and everywhere. And finally, that CO2, that's basically 20% of the CO2, begins binding onto the globin group. And finally, to keep it in place, because you don't want the CO2 to just start detaching a little bit everywhere, you want it to be in place, you need a seat belt, basically. And that seat belt is going to be your 2,3-DPG, which stands for disphosphoglycerate, 2,3-diphospho or bisphosphoglycerate, or glycerol, however you want to say it. But anyways, 2,3-DPG travels towards and goes inside of the red blood cell and binds on to the hemoglobin to keep everything in place so that the CO2 doesn't detach. That as well contributes a little bit to the whole process. And then finally, I didn't really say what happens to the 70% of the bicarbonate that just got converted. Basically, as you can see, there's one last arrow. It's the hamburger effect, but in reverse. Basically, HCO3 goes inside of the into the circulation inside of the plasma, which in return intakes by co-transport. By exchange. Chloride anions, basically, inside of the red blood cell to prevent the lysing of the red blood cell, because too much of an acidic pH can cause the lysis of that red blood cell, and it's very dangerous. You don't want that. And that's basically the Bohren-Haldane effect, and how the exchange between CO2 and O2 happens, and the exchange between the capillaries happen, and the transport of oxygen and CO2 throughout the body happen, in a nutshell. Finally, we get to our blood pathologies. I'll go really quick into them because I really want to show you the microscopy video. We're going to start with anemias. Anemia basically means that you have a lower level of red blood cell. General symptoms of these guys are: you're going to have shortness of breath, or dyspnea, or like difficulty breathing, which is dyspnea. A lot of fatigue because you're not receiving a lot of oxygen, not a lot of energy being produced as well, not a lot of ATP being produced. Tachycardia, and that's really interesting because when your heart senses that you don't have a lot of O2, actually it regulates itself with the partial pressure of CO2. If there's not enough CO2, it begins to think that there's not enough oxygen, and it starts pumping a lot faster. You'd have to go and check out the cardiac system, cardiovascular system videos in general to see how that occurs, because we're not going to get into that today. And finally, an increase of cardiac charge. Because of that, the heart begins pumping a lot faster because it wants to deliver that oxygen that's missing throughout the body, logically, when that happens. So these are a few of the general symptoms. One of them, of the numbers of anemias that are occurring inside of the body, we'll start with the iron deficiency, which is probably one of the most common ones. Causes can either be the blood loss. You're going to see that anemias also include blood loss inside of them. Menstruation, or a decrease of iron inside of the diet. The treatments for this is an increased iron intake, either food or by supplements. And if it's like really, really bad, it's a transfusion in the last resort. So as you can see, usually what occurs is there's either an iron missing, and that iron is just missing here inside of the red blood cell. It's really straightforward. So a decrease in iron means decrease in heme. A decrease in heme means a decrease in your hemoglobin, basically. You could also have a decrease in vitamin B12 or folic acid, depending on what you eat. This one is mostly called pernicious anemia. pernicious anemia is the synonym of your vitamin B12 or folic acid decrease. As we saw in the video of your erythropoiesis video, the vitamin B12 is used for the growth of that red blood cell. Basically, this one is like a sort of immune response attack. It's an autoimmune syndrome because your immune cells begin attacking the vitamin B12, or they can also attack, same thing for the folic acid, they just attack it. Basically, these immune cells attack your intrinsic factor, which see it as a threat. They secrete this antibody that binds on to the intrinsic factor and takes its place, meaning that the B12 can't bind on to the intrinsic factor anymore. And as we saw in the erythropoiesis video, intrinsic factor is used to transport the folic acid into the bloodstream in order to help the growth of the not the growth, but more of the maturation of the red blood cell. And what happens is that without B12 basically compresses your red blood cell into this shape. But without B12, you're going to have yourself with a really, really large red blood cell like this, and it's called macrocytosis. It's a macrocytosed red blood cell, which is unable to mature the red blood cell and produce DNA synthesis for the red blood cell. Treatments for this include an increase of vitamin B12 by intramuscular injection, because you wanted to have access as quick as possible, to go as quick as possible to anywhere towards the red bone marrow. You don't want it stagnating into the stomach, basically, because that's where your immune cells start attacking your intrinsic factor, you know? We move on to aplastic anemia, which has a synonym called idiopathic anemia. It's basically the myeloid stem cell that there's a failure with your inside of your red bone marrow, where your myeloid stem cell just stagnates in this form. It doesn't go towards the platelets, towards the red blood cell, or the white blood cells, you know? It's an issue with the myeloid cell division process. If I'm not mistaken, the scientists are unsure what causes this, but the only treatment for this is a red bone marrow transplant. Moving on to the glucose 6-phosphate dehydrogenase. That's really a mouthful to say, but the G6PD deficiency. Other symptoms other than the general ones included here, they include other symptoms: dark or yellow orange urine, or jaundice. Jaundice is the yellow pigmentation of the skin or the face. You can generally see it on the face or at the tip of the fingers, the skin as well, but mostly you're going to see it on the face, especially in the eyes. You're going to see, like, if I'm mistaken, it's icters for the eyes. You'd have to correct me on that, but I think that's what it is. And the reason for that is that the G6-GPD contributes to the heme functioning correctly. What happens in a normal reaction is that the glucose with the G6-GPD produces the heme. It is essential in that heme production pathway. If there's a lower if there's a lower G6-PD, there is heme insufficiency. That means your heme is not going to function as well. If you have heme insufficiency, that means it's not going to bind on to the globin correctly, and you're going to have an increase of Heinz bodies. It's what that's going to cause, and those Heinz bodies is going to contribute to the lysing of the red blood cell, and this is where your jaundice and your dark yellow urine come into play. If you remember, when you lyse the red blood cell, it has to get digested one way or the other. If you lyse too many blood cells, you have an increase of bilirubin inside of the bloodstream, which leads to an increase of urobilinogen, and that's what kind of causes the jaundice. Actually, the unconjugated bilirubin is going to cause the jaundice. The dark or yellow-orange urine is from the lysing of those red blood cells because they eventually go inside of the kidneys, they get filtered, and that increase of number one, of urobilinogen or urea, once again transformed into urea, that increase in particles of the red blood cell contribute to that dark yellow-orange, you know? Now, the treatment for that, it could either be antibiotics if it's caused by an infection, supplemental oxygen in severe cases, and transfusion in severe cases. Now, there's also sickle cell anemia, otherwise known as falciform anemia. Other symptoms can include, it can also cause vasoocclusion, because this specific shape, what happens is that this is more of a genetical cause. It's a genetical mutation that causes, if I'm not mistaken, the valine at the 6th position of the beta chain. Yeah, the beta chain. The glutaric acid is replaced with valine at the 6th position. There we go. It was right in front of me. And what causes it is to have this shape. And if you remember, hemoglobin needs to have this like squared shape versus this stacked shape right here. They begin stacking on top of each other, which creates this sickle shape of the red blood cell. And this sickle shape of the red blood cell, eventually it's really dangerous because they can clump together to create vasoocclusion of inside of the capillaries, which is really dangerous because you could eventually get like occlusions, stops, blockages, anything else that's really dangerous. Treatments for these include hydroxyurea, opioids for pain relief. These guys are in a lot of pain once you get a lot of occlusion going on. Fluids to help more fluids to actually help decrease the viscosity inside of the blood. And then finally, supplemental oxygen in worst case scenarios. We move on to thalassemia. Thalassemia is, if I'm not mistaken, it's really common to the Mediterranean's individuals. It's basically a deficient or missing either B chain, a deficient or missing alpha chain. Usually it's by genetics, and the treatment, honestly, for that is just more iron intake or food supplements. It's just that you won't have enough space for another like chain, and you're not gonna have enough oxygen going through. But it's not that bad compared to the other ones that I've just explained. Finally, there's I think one more to go before we end up with the last one. We have hereditary spherocytosis. Other symptoms include splenomegaly, and you're going to see why is because the causes is a genetical mutation that causes the ankyrin, and I've said it before at the beginning, ankyrin is mostly used for that flexibility of the red blood cell. The ankyrin, which is binding on to the spectrin, the mutation affects the spectrin protein, and the problem is that if spectrin doesn't bind correctly to the ankyrin, that means that your red blood cell equals that it's going to be more, number one, more spherous. It's not going to be as flexible to go through the capillaries, and it happens that it's going to get stuck, which is why it causes splenomegaly. Splenomegaly, which is an increase of the spleen, an increase of the shape of the spleen. Other treatments, some treatments include transfusion. Basically, and it's the only treatment so far to give normal red blood cells, a normal shaped red blood cell, basically, to the patient. Finally, like I said, one of the anemias include hemorrhagic anemias. If you're losing blood, you are technically in anemia, in an anemic state, because you don't have enough red blood cells to give oxygen to the tissues. Again, it causes heavy blood loss, and the treatment is transfusion. It's basically as straightforward as that. And then finally, we end up with other blood pathologies, which one of them is, I'll start with the first one, it's just the hemolytic disease of the newborn, otherwise called Erythroblastosis Fetalis. This has to do with the blood typing that we saw earlier at the beginning, which is really important to that I've explained. Basically, it's the it has to do with the fetus, with two fetuses actually, and you'll see why. When the mother has the first pregnancy and has a first fetus, that first pregnancy, if that baby just so happens to be Rh positive, either from the father, or actually it has to be from the father, if I'm not mistaken. It has to be a genetic mutation. Well, it has to be genetics into play as well. But in any case, the baby is Rh positive, and the mother it's Rh negative, and that's where the Rhesus factor, which was dangerous a little bit in this pathology specifically, is really dangerous, because that Rhesus positive blood transitions inside of the mother's blood circulation. It gets exchanged and then travels, and then what happens is the immune system starts producing anti-Rh antibodies. But the good thing is that nothing dangerous actually happens to the first fetus inside of the first pregnancy, because first of all, to produce these antibodies, it takes time, so it's not going to be that dangerous at the first pregnancy. What does happen is eventually at the second pregnancy, if again the mother has another baby at the second pregnancy and the baby happens to be Rh positive, now the mother becomes Rh positive and has these antigens, not the antigens, but the antibodies inside of her circulation. It crosses over into the baby's fetal circulation. And what happens is, like I said before at the beginning, these antibodies cause the aggregation of the red blood cells, which lead to hemolysis, anemia, decreased oxygen, and potential death for the fetus. The second, like I said, in the first pregnancy, nothing happens. The baby is really normal. At the second pregnancy, because there was a lot of time to develop these antibodies, with the second pregnancy, the baby, the issues with the infant at the moment that he gets born is anemia, an increased amount of bilirubin, icterus or jaundice in this case, and then slowdown of mental development. The treatment for this is really, there's one treatment so far that exists, because you have to catch it fast. The early the better, and what you do is that they prescribe this medication called Rogam to block the antibody, to slow down basically the mother's immune system from synthesizing antibodies, and to also block the antibodies from interacting with the baby's Rh positive red blood cell. And then finally, another one of the blood pathologies, there's actually two of them, but they're the same one. It's polycythemia, or in this case, we're going to look at first the first degree polycythemia, which is the first degree polycythemia is actually an increase of red blood cell synthesis. Symptoms of an increase of red blood cell synthesis include an increased blood viscosity, which can lead to thrombosis and or embolism, and a high increase of bleeding. What happens is the mutation inside of the myeloid stem cell, there's this JAK protein, which stands for Janus kinase. That also is responsible for the synthesis of stimulating the myeloid stem cell to mature into the erythrocyte. What happens is that there's a mutation with the JAK protein, which causes to always occur to always go towards the erythrocyte pathway, even if there's enough red blood cells in the circulation. Even if there's enough red blood cells inside of the circulation, it's still gonna go through this pathway, and you're gonna have way too much red blood cells in your system, because the JAK protein is the one responsible for controlling if you have enough or not enough red blood cells. It actually decides, am I gonna go through with this pathway towards the mature erythrocyte and have red blood cells in the circulation. And that's what basically happens. Finally, you got secondary polycythemia inside of the nephron cell. This one is an too much increased amount of EPO. That basically your hypoxia-inducing factor stimulates itself, produces too much EPO, which leads to too much mature erythrocytes plus, basically. Triggers for EPO is basically hypoxia. Hypoxia, when you don't get enough oxygen towards the tissues, your body thinks that I need to produce more red blood cells. Higher altitude, which triggers it. Cardiovascular pathologies, which triggers it. And finally, it could be renal cancer, which makes it there's renal insufficiency towards a cancer, which overstimulates this production of EPO. So that was basically it for the other pathologies of the blood. Finally, here are my references. As always, big thank you to Ninja Nerd. If you really want to take a look at his video, he goes really much in precision to every single aspect of what I just explained right now. And finally, the main part, thank you for sticking so far with me. Finally, we're going to go to the main part of this stream presentation, and we're going to take a look at the erythrocytes. Now, this was taken a while ago. It's like pre-recorded. But this is a blood sample basically that I took. Don't do this at home, please. Don't prick yourself. I had to prick myself to showcase towards on the microscope and to showcase what happens. But this is at 100 magnification. As you can see, the blood is like really. This is what you're seeing as well. This is 400 magnification, if I'm not mistaken. I'm trying to look at to see if I can find at this point really good red blood cells. As you can see, these are clumps. This is what kind of happens when you prick yourself. You're just going to get these clumps eventually of these red blood cells. And this is actually really interesting. This is what I was talking about for the... I'm just going to reverse it a little bit. And I'm going to wait for... Yeah, as you can see here on the circle, I just paused the video. On the circle over here, look at this red blood cell on how it flexes. It flexes its shape. That's the ankyrin and spectrin protein into play. Look at how it literally like flexes. If you want, you can actually see it again. You also have it at the bottom left corner. You can also see the red blood cells just like changing their shape, flexing together. It's really, really interesting. It's really interesting to see under the microscope when that occurs. So basically, if I go back to what occurs with the clumping, what occurs to the stacking up of the red blood cells, as you can see here, is, if I'm not mistaken, it was the coagulation. That's what happens when they're, like, outside. That's how they coagulate together, you know? That's about it I can tell you about the stacking. I'm not sure why they stack. And here you can actually really see it clearly. You can clearly see the changing of the flexing of the shape, how it flexes, how the red blood cell flexes its shape to go. And that's basically what occurs in tight spaces in the capillaries for the red blood cell, you know? Aside from that, what else? Yeah, here you can also see them clearly. You can see them going around at the bottom left corner. You can actually see the red blood cells flexing their shape. Look, there's more of them coming at the same spot. It's really interesting to look at that, to see how that ankyrin and spectrin protein work together to allow the flexibility of that red blood cell. It's really, really, definitely interesting to see. And this is the stacking that I was talking about. Here you can actually see a red blood cell by itself, or multiple red blood cells by themselves. One of them at the left, and there's another cell. I'm not sure what it is. I'm honestly not sure what it is, what that was. I'll just go a little bit back to it. There's a cell over here. I'm not sure what it is. Next to the to the red blood cell. I'm not sure if that's an eosinophil or a neutrophil. You guys can leave your comments inside of the comment section to let me know what the one next to the red blood cell is. If it's a red blood cell. To let me know what it is. And here we're looking at red blood cells basically moving together inside of the fluid. Because what I did is that, yeah, you have when you prick yourself, yeah, you just give a little bit of your blood as well. Oh, and look at that. Those are perfect, actually. Those are perfect flexions of the red blood cells, as you saw. I can actually, like, bring it back. Look at that. Look at how they drop and they just flex. That's really amazing. I think that's another cell. I think that's a macrophage, if I'm not mistaken. I'm not even sure. That has to be another cell that's touching together. I'm not sure what it's touching, but it's also in movement. Don't remember what I was saying previously, but that really caught my attention right there. After that, I'm not sure what I'm actually trying to find here. I think I was trying to find if I could find like white blood cells as well. But anyways, when I pricked myself, I didn't just put blood on the slide. I also put what's it called? Water as well. And that's why you saw the red blood cell pulsing through the water like that, you know. This one that I circled, I'm not sure what it is, but it seems to be another cell. I'm not even sure if it's an immune cell, an immune system cell, because it's gonna move. It's gonna move a little bit. You could clearly see it moving its shape. If I as you can see, it's like literally moving its shape. I don't even know what it was. I thought it was an immune system cell, but it doesn't have the shape of an immune system cell, you know. It may or may not be a macrophage. I'm not really sure. But that was really interesting to look at. As you can see, it just changes its shape. And if you want, I can also like do advanced like this. As you can see, it like changes its shape. And if I reverse, you can see it. You can see both of them changing their shape. As you can see. And I don't even know what this type of cell is, but if you guys can like let me know what it is in the comments, I'll highly appreciate if you tell me what this type of cell is in the comments. Look at that beautiful cell moving. I think that was basically the end of the video. Yeah. Well, now the video just basically restarted. And this was it, guys, for the red blood cell presentation. I hope you enjoyed it. I hope it was really instructional. I tried my best inputting as much information as I could within the hour and 30 minutes. I'll probably cut it, edit the video out. But in any case, thank you very much for being part of this stream. Like I said, if you want more notes from lecture notes, they're available on Patreon for high quality print. If you would like to have them to maybe help you throughout your studies or something like that. In any case, thank you very much for watching. Please hit the subscribe button. Thank you to have made it so far in this stream. Hit the like button, the share button, and I will see you folks for the next science video stream.
Hey guys, thank you very much for watching the whole content. I hope you enjoyed it. If you want to catch it live, come on twitch.tv/MajinP, that's MAJINNP. I appreciate the support this channel has been getting, which is making it growing bigger and bigger. So please take the time to like, subscribe, and share, and all that fun stuff. I'm always here trying to get up there, so if you want to support your boy, you know what to do. Thank you very much again for watching, and peace out.